Hello, my name is Crystal Reyes and I'm from Lone Star Honors College in Kingwood. And today I did an experiment with tiny earth over antibiotic-producing soil response to non-producing bacteria. So, I want to bring your eyes to this number, 35,000. What's so significant about this number? Well, according to the CDC, 35,000 people died in 2019 due to antibiotic-resistant infections. According to that same statistic, 2.8 million people contracted antibiotic-resistant infections every year prior to that one. So, that led me to my research question. How does antibiosis affect the relationship between non-producing bacteria and antibiotic-producing bacteria in the same soil sample? So what is antibiosis? Well, according to the Oxford Dictionary, antibiosis is an antagonistic relationship in which one microorganism is adversely affected. So to put that in a visual sense, this is a sign of antibiosis. So you can see in this media plate right here, there's all these dots around them and some are touching. However, the one in number 5 is not being touched because the bacterial colony over here is producing a substance that is pushing away all the bacteria that is taking over the media plate. So for the reason why I did this experiment is because I'm working with tiny earth to find an antibiotic producing bacteria against the escape pathogens. Now, the escape pathogens are some of the bacteria which lead to those 35,000 deaths that I talked about. Some of them being Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. So for the experiment, we had to work with the safe relatives to, of course, avoid infection. So some of those being B. subtilis, as the one top there that I showed before, the other one being E. raffinosus, E. coli, E. acrogens, P. putida, S. epidermidis, M. smegmatis, E. cartova, and finally A. bailey. So the materials that I used for my experiment are the inoculum loop, as you can see right here, of the glue that the hand is holding. And then we had a container for soil selection, which you'll see on the next slide. Then we had the incubator to, of course, incubate our soil samples and the bacteria. And then we had a micropipette. And you also had the media plate over here, which is filled with yellow gel. So for the beginning of isolate collection, little known fact about our antibiotics is that 95% of them come from the very soil that we stand on. So for my experiment, I had to collect 2 different soil samples from 2 different locations, and then further use dilution to isolate bacterial colonies to make a library plate, as you can see the very distinct colonies in these 16 quadrants. Now, for the real experiment to test whether it would react with each other or not, I used the cross-streak method. It's a method where you use the inoculum loop, as I've shown before, to streak across the media plate against the antibiotic-producing bacteria. So first, you would streak your antibiotic-producing bacteria, and then you would go across and streak the same colony, and in my case, it was a non-producing bacteria. So for my experiment, it looked a little bit like this. So you can see that while some of these do touch, that is not an interaction between the bacterial colony and the antibiotic production. But however, for isolate number 3, you can see that it's still being pushed away by the antibiotic producing bacteria. And you can see the same for my positive control as well. So for my first experiments, I used the incorrect method of cross-streak. I thought that you would have to place the isolates on one side only and not touch the line to indicate antibiosis reaction. However, I was incorrect. Still, I still got results from this experiment as you can see, these three different series, the RP series of these three, the CR series over here, and the BGP series with isolate number 5. Now for my negative control, it was these three different isolates, the positive being with L. antibioticus, a known antibiotic-producing bacteria. And then we have my experimental control group, which had the antibiotics from the very same soil sample as my non-producing bacterial colonies. So for non-producers with different soil samples, you can see the interactions. As for over here, the bacterial colony is being taken by the antibiotic-producing bacteria, but those colonies are from different soils, so there would be an interaction nonetheless. And these are all from these isolates over here are all from different colonies, and this one is from a different soil sample entirely. Meanwhile, for the plate over here, these are all from the same soil sample. And as you can see here, the yellow colony, isolate number 3, still touches the antibiotic-producing bacteria. So therefore, there's not a real sign of interaction within it. And for the rest of my experiment, unfortunately, it did look like the rest of these plates. There's the plate I shown before, and the rest of my experiments, you can see that they still touch the antibiotic-producing colony, meaning that there is no real interaction shown between the colonies and the antibiotic-producing bacteria when of the same soil sample. So the significance of this experiment was to help identify interactions between microorganisms of the same soil sample and to further the understanding of antibiosis, as we still don't know the reasoning why antibiosis truly happens. And for further research, I would like to know what causes antibiosis, as well as what the cultural characteristics of these isolates are, as I don't know what these isolates are just yet, and I don't know whether they are a known antibiotic or a new one. And thank you.